colony PCR is a quick and easy way to tell if your cloning probably worked without having to go through like a mini prep. And this can be really helpful if you have a ton of colonies that you're trying to check which of these colonies were cloned properly. That is, they have like the insert, so the genetic information I wanted to put into the circular piece of DNA called a plasmid. Basically, you want to make sure that each of the bacteria, these different colonies, are genetically identical bacteria that kind of like grow on top of each other. And some of these are going to have the plasmid, some of them are going to have a wrong plasmid, or I mean like your sequence wasn't actually in there, some of them the insert might be in there incorrectly, and so you want a way to check whether the insert is okay. And the only true way you can do that is with sequencing, and you'll still do that later, but you don't want to have to go through and sequence all these different colonies in order to find out, well, which of these ones are actually good, could I actually use? And so colony PCR is a quick and easy way to do this. Here's basically what you're going to do is you're going to take a pipette or a sterile toothpick or something. You take you take a colony and you kind of just touch it really quickly, and then you swirl it around in your PCR mixture. Now what happens in PCR is that you're going to make lots of copies of a defined region based on a template. And so in this case, the template would be the plasmid. And your plasmid is going to have like the vector backbone, which is kind of the generic part, as well as your insert, which is the part that you put in there. Now in PCR, the region that you amplify, that you make copies of, you determine the region kind of like by bookending it by these little pieces of DNA called primers. You can design these primers to either be inside the insert or inside the vector, and depending on where you place the primers, you will get different sized products or you won't get products depending on whether or not your cloning actually was successful. And so I'll get much more into all of these details, but basically PCR is going to be able to tell you whether your insert is probably in there. And so normally when we run PCR, we're running on purified things. And so we could do a mini prep and basically grow up these colonies in liquid media, then purify out the plasmid, then do the PCR on the purified plasmid, but we don't have to go through all that work with colony PCR, because the PCR, when it heats up in the PCR cycling, it gets hot, and when it gets hot, it's also going to break open or lyse these cells. And so if we just take a little bit and we swirl it around in that PCR mix, during the PCR reaction, when it heats up really hot, the DNA is going to get exposed to all of the DNA copying machinery. And so then if your insert is in there, then you'll see products of the expected size when you run an agarose gel. And so much more in the details in a minute. At the same time as you do that PCR, you can also be kind of like starting a 5 ml overnight culture for a mini prep. And this way, if your colony PCR was successful, now you can go ahead and you already have that culture growing for your mini prep, because you're going to need to purify out that plasmid now, but you're only going to want to have to purify out the ones that have a hope of actually having your colony of insert, like your plasmid that you want. And then you can go ahead and send that for sequencing. So now let's step back and get into all these details. Now I give you this like broad overview of where things are going. In molecular cloning, we can take the genetic instructions, say for making a protein, and we can stick them into another piece of genetic information. And so often what we're doing is we're sticking a gene or like an insert, so basically the cDNA, the complementary DNA of the RNA of the instructions for making a protein, basically just the instructions for making a protein. We can stick those into a circular piece of DNA called a plasmid, and then we can stick this plasmid into bacteria. And bacteria are going to make lots of copies of that plasmid and keep making the plasmid when they divide. So basically you have this kind of endless supply of this plasmid, and it's really, really awesome as long as that plasmid actually has the sequence that you think it has. So we can use techniques like restriction cloning and SLIC in order to actually get these plasmids made, but these techniques aren't perfect, and sometimes it might not work, and you might end up with plasmids that aren't the right thing, and you don't want to be feeding the bacteria and using this plasmid that isn't the right thing. So we have different methods in order to make sure that the plasmid is what we think it is, and that our insert, the gene or the genetic instructions we put in there, are really in there, and they're in the right orientation, and they have the right spelling, there's no typos, all those various things. The only truly way to definitively tell if all that is right is to actually sequence this plasmid. But in order to do that, you have to go through some purification, and you have to go through various steps, and it can be a pain, it can be time consuming. Not that it's really hard, it's just time consuming and boring, and then it's resource intensive. You have to pay, you have to wait. What if you just want a quicker and easier way? Well, colony PCR can't tell you for sure whether the sequence is right, but it can give you a good idea whether your cloning worked. And how it works is basically what you do is when you do this cloning, you get a quick way to see if the plasmid actually got into the cells by antibiotic selection. So we have a couple of different steps at which we're kind of doing quality control. So when we do that molecular cloning, we're basically making this plasmid, but then we need to get that plasmid into bacterial cells. And so this is a step called transformation. And sometimes the transformation doesn't actually work well. And so you're left with bacteria. Some of the bacteria don't even have the plasmid. And so you only want to like grow the bacteria that have the plasmid. And so what you do is in your plasmid, you have an antibiotic resistance gene. And this is going to act as a selection marker. So basically, you grow the bacteria in the presence of that antibiotic, and only the bacteria that actually have the plasmid will have that antibacterial resistance gene. So only those cells will be able to grow. And when they grow, they're going to grow in these little kind of clumps. Basically, a bacteria will take root and it'll keep copying itself, duplicating itself on top of itself. And you get these kind of gunky, globby things called colonies. And each of these colonies should represent a genetically unique clone, or, I mean, they should, hopefully they should actually all be the same with your plasmid in there. But basically, each of these is going to be a colony, a population of bacteria that are the same. And so some of these will have your these should all have your plasmid because you used antibiotic selection. You hope that they all have your insert, and you hope that the insert is spelled correctly in all of these, but it might not be. And especially with some cloning methods, especially if you're having difficulty with your cloning, some of these really might not have your sequence, your insert in there. And you're not going to be able to know based on just the selection because the selection is coming from the plasmid, from the backbone, and not from your insert. And so we need a way to tell if the insert is actually in there as well. What we can do is we can take the colony and we can actually purify out the plasmid. We can do this using a technique called a mini prep, where basically we grow up, we take a colony and we take a toothpick, or we take a sterile like thingamabob, and we basically touch the colony and we stick it into about 5 mils of LB, lysogeny broth, basically bacterial food or media. And then we can stick this in the shaker incubator and we grow it overnight to get lots more copies of that plasmid. And then we go through these steps where we purify out the plasmid. You have these like kits and you have these columns, and it's pretty easy, but especially if you're trying to test a lot of colonies, it can be a real pain. And so vacuum manifolds can speed this up, where instead of spinning to get things through the column, basically you have a vacuum. So how these columns work is they're going to kind of bind to the DNA and then everything else flows through, and then you do a bunch of washes and stuff. So you end up walking back and forth to the centrifuge, spinning over and over. And if you have a bunch of tubes, this can be a real pain. And so basically colony PCR helps you not have to waste your time doing that for the bad colonies. And furthermore, because you're going to go straight to the PCR without having to do that purification, and you don't have to waste your time actually going and sending these colonies for sequencing, or even just that sort of thing, if you know, you kind of have an idea that they're not what you want. And so how colony PCR works is you basically take those colonies and you run PCR directly on that goopy doopy dot. And at the same time, you can start one of those overnight growths. Because if the colony really does have what you think it has, if the PCR reaction was positive, if it looks like it's right, then you want to kind of have a step ahead, be like one step ahead where you already have that culture growing of the bacteria, so that you can do the mini prep the next day. And you don't want to have to then be trying to cross-reference back, okay, which colony did I take and stab from before? Which one corresponds to which PCR product? And you don't want to have to be digging back into the same colony and have the potential of contaminating things. So at the same time that you prepare your PCR sample, you can prepare for the mini prep. And so typically what I do is for this PCR sample, I just barely catch that colony with a sterile toothpick or one of those like more official tools, like one of those plastic swabby things or a pipette tip. Basically, you just take it and you scoop a little tiny bit of the colony, and then you swirl it around in your PCR mix. And we'll talk more about this mix in a minute. Alternatively, you can resuspend a bit of it. So basically pipette it up and down in some water and then add this to your PCR mix. Now, when the reaction heats up, so in PCR, the reaction is going to heat up to melt apart the strands of DNA. And when it does this, it's also going to break open or lyse the cells. So you don't have to worry about doing the cell lysis yourself. And so the reaction is going to basically lyse the cells for you. So the DNA is going to spill out. Then DNA polymerase, so the DNA copier, can latch on and it can make lots and lots of copies as long as the sequence is there to copy. If the sequence is not there to copy, then you're not going to get products, and this will tell you that your product isn't there. Or it also depends on which way the primers are designed. And so we'll get more into this. But basically, the presence and the size of the products can tell you about whether or not the insert was there. And because at the same time, you took that one swab and you stuck it in the PCR mix, you took another swab at the same time, and you stuck it in your 5 mil growth. And now you have the sample where if your PCR product worked, you can go ahead and the next day, because you do this growth overnight, typically the next day, you can go ahead and mini prep it, send it off for sequencing to make sure everything is really okay. Or if the PCR product was a dud, then you can just bleach and toss this media. And so you don't waste time actually mini prepping it if it's not something that you're actually going to need. So let's talk more about the design of this PCR. The idea is kind of similar to how tests like molecular diagnostic tests work, such as the coronavirus tests, those PCR tests. Basically, PCR can make lots of copies of a sequence. If the template sequence is present, it can make lots of copies of a region of that sequence. And the way that you make these lots and lots of copies is you specify the kind of bookend the regions that you want to be copied using these short pieces of DNA called primers that kind of act as start points for the copying machinery, DNA polymerase. So you have one primer per strand, and this DNA polymerase is then going to copy, and it's going to copy until basically the first cycle, it's going to copy till it runs out of steam. Then the second cycle, it's going to run till it's going to kind of like fall off where the other primer had started. And then all the future cycles, you're going to get pieces bookended by the primer start sites. And much, much more on this in other posts. But basically, this is happening in these cycles where you have the DNA strands coming apart, the primers latching on, the DNA polymerase doing its job, doing that copying, and then those new copies separate. And so every cycle, you're going to get more and more and more copies. But this can only happen if the template sequence was there to copy in the beginning. The sequence was there that those primers recognized and bound to. And so in colony PCR, we can basically use primers that bind in the insert and or the vector in order to figure out whether or not the insert was in there. And if we use primers that are in orientation specific primers, we're also going to be able to tell whether the orientation was correct. So this isn't necessary for all, this isn't an issue for all cloning methods, but for some cloning methods where you have like blunt ends, what can happen is this insert can actually go in be backwards. And if you insert in cloning things, like if you just had vector specific primers, you wouldn't be able to tell, but we'll get into that in a second. So what we're going to do is we can design primers that are going to work in different ways. And each of these has pros and cons. So the orientation specific primers are going to give the most information, but they're also going to be the least generalizable in terms of being able to use the same primers with different reactions. So in order to talk about what I mean, basically let's think about where we put these primers. If we use vector-specific primers, so we have both of our primers. Remember, we need two primers, one that kind of specifies a start and one that specifies a stop. If we have both of these primers in the vector, then what's going to happen is that if they're going like into the insert, If the insert is in there, you're going to get a big piece. If your insert is not in there, then you're going to get a smaller piece, or it could be a really small piece depending on how close together your primers are in the vector. But now you don't have an insert there, and so this region is going to be missing. You'll get this short little thing. And if it's facing the wrong way, well, here you would still get the same size pieces if it were facing the right way, because nothing is specific. Your primers are only recognizing the vector and not the insert. A benefit of vector-specific primers, though, is that you can use, often what you're doing is you're subcloning different inserts into the same vector. And so in this way, you're able to you'd be able to use the same vector-specific primers with different inserts. Another option is insert-specific primers. So here, both of your primers are recognizing sequences in the insert. A benefit of this is that you can use it for the same insert in different plasmids, vectors, but a drawback is that you don't know if your insert is actually where you want it to be. You just know that the insert is somewhere in that mixture. And so if the insert is in there, you'll get this PCR, you'll get these short products. If it's not in there, you won't get any products. But again, this doesn't mean that your insert is actually where you think it is in the plasmid. Orientation-specific primers are going to give you the most information. Here you have one primer that's in your insert and one primer that's in your vector. If the insert is there and it's in the right orientation, then you're going to get a product of the expected size of kind of like your insert and part of your vector. If the insert is not in there, well, now this one primer is not going to have anywhere to bind to, and so you're not going to get any product. And if the insert is in the wrong direction, well, now your primers are both facing the same direction, and so you're not going to get any product. And so this is why I say that the orientation-specific primers are going to give you the most information. But how do you actually visualize these products? We can visualize these products using an agarose gel electrophoresis. So basically you have this sugar mesh gel, and you're going to send these DNA pieces traveling through the gel. And they're going to separate based on their size, because what's going to happen is that the longer pieces are going to get tangled up more in this mesh in their way through the gel. And so what's going to happen is that those pieces will going to travel less far. And so when you turn off the machinery, basically you remove the electrical drive that's pulling the DNA through the gel. Now those pieces are going to be stuck in place, and the bigger PCR products are going to be higher up. The smaller PCR products are going to be lower down. And then you can compare them to a ladder, so these like molecular weight standards where you have these pieces of DNA of known size, and then you can compare them and see whether the pieces look to be the size that you would expect. So if you were to use vector-specific primers, you're going to be looking for that big piece that has that goes all the way through your insert. If you use insert-specific primers, you'll get a smaller reaction product corresponding to the insert or part of the insert, depending on where you place your primers. And if you use your orientation-specific primers, then you're going to get a big piece, or it depends on how big it is going to depend on where you place your primers. But you should only get a piece, you should get a piece that corresponds to your part of your insert and part of your vector. And this is going to give you the most information. But you're going to need a primer that's specific for both your vector and a primer that's specific for your plasmid. This is going to tell you if your insert is okay, but not if that sequence is okay. And in order to tell if the sequence is okay, then you're going to have to send it off for some sort of sequencing. So in this case, what you would do is you would actually do your mini prep to purify out that plasmid, and then you can send it for sequencing. There are a couple of different options for sequencing. What I've been doing lately is like whole plasmid sequencing. It's now like basically not very expensive. You can get your whole plasmid sequenced. Traditionally, what we would do is like Sanger sequencing. Basically, you could design a single primer that would be starting, or you could do multiple primers if you wanted to. Normally, I would do one on each end so I can kind of get the sequences of both of the places where the cloning took place, because those junctions are going to be the most prone to errors. But basically, you design these primers, and then in the sequencing reaction, it does kind of like PCR, but it only does a single, like you only have a single primer, and it's just going to copy a strand of DNA and read it out as it does so using these like fluorescent dyes typically. And so with Sanger sequencing, you're able to then get the sequence based. You send the primer. This could be a primer that is specific. Often these kind of like generic vectors, they have sequencing primers that are like standard sequencing primers. And so the sequencing facility will actually have the primers for you, and you just have to tell them which ones to add, or you can have custom primers that you would like premix in there. And then they'll run the reaction and they'll sequence the DNA and send you this sequence. And alternatively, you could just send the whole thing for the sequencing. When you get this back, you want to basically check your traces, more on this in other posts, but you really want to check those junction points, the places where your insert meets the vector, which is where you're most likely to have typos be introduced. And so the colony PCR is a way to kind of choose which ones you're going to like prioritize which ones you want to actually send for sequencing. So you don't go through the mini prep, you don't go through the sending for sequencing, don't go through all that stuff for colonies that don't actually that aren't likely to have the right thing in the first place. Another way to check if another similar type of thing to colony PCR is like a restriction digest, or instead of making copies of specific regions, if the insert is in there, you actually can make cuts if the insert is in there. And so much more on this in another post. I prefer colony PCR because you don't have to do the purification beforehand. With the analytical digest, you can do this before you send it to sequencing, but you still would have to do that mini prep and things like this. If you want to be even lazier and see, okay, well, is my insert probably in there before I even go through the colony PCR, you can use some sort of screening method. So basically, we can talk about selection versus screening. In selection, basically what you're saying is only the clones that have some sort of property, only the clones that have something will be able to survive. We're going to select for those ones. We're going to select for the ones that have the plasmid with the antibiotic resistance gene. In screening, basically you're going to flag the ones that have something or don't have something, but you're not going to actually like remove them. Like we're not going to kill the colonies that we don't like. We're just going to show which ones are probably good and probably bad. A common way that we do this is blue-white screening, or basically when we put in the insert, we do it in the middle of this gene that's needed to make this blue product if we grow these on special plates. And then if our insert gets in there, basically it can't make the blue product. And so you get this white colony, and then you prioritize these white colonies for your colony PCR, for your analytical digest, for your sequencing, for whatever you're going to do. So to review, basically in colony PCR, you just take one of those tiny little colonies, you stick your PCR tip, or you take your pipette tip, your toothpick, something like this, take a little swab of that colony, mix it around in your PCR mix. When that PCR mix heats up, going to break open those cells. When those cells break open, now they're going to come into contact with those primers that you've designed to either like be in your insert or your vector or one in both. Those primers are going to bind. If your insert is in there, or if depending on what primer sequences you have, basically you'll either get different products or you won't get products at all, depending on how you design your primer and whether or not the insert was actually in there and whether it was in there correctly. And so then by analyzing the size of the pieces with agarose gel electrophoresis, you can then see if you get the products of the expected size. If you do, then what you can do is you can go ahead and do that mini prep, purify out that plasmid, and send it for sequencing. Then check the sequencing to make sure things are really okay. And now you can go play with your plasmid. So hope that helps save you some time and helps you understand colony PCR. It's a huge, huge, huge thumb and time saver.